Hi, my name is Kira Miller, and I'm a first-year Integrative Biology Master's student in the Vredenburg Lab at San Francisco State University. My research interests stem from several seasons of studying amphibian populations in the Sierra Nevada, most notably the endangered mountain yellow-legged frog. Imagine you're out hiking in the mountains when you walk up to a lake to see this: frogs living in lethargic or dead. Now imagine that you had been hiking in this very place just a few years before, and at that point you saw frogs, but they were abundant and thriving. This is what a BD die-off looks like. BD, or Batrachochytrium dendrobatitis, is an aquatic fungal pathogen that infects amphibian skin and leads to the disease chytridiomycosis. BD has caused global amphibian declines, but mysteriously does not affect all amphibian species in the same way. In California's Sierra Nevada mountains, we have two species of frogs with very different susceptibilities to BD. The mountain yellow-legged frog, Rana muscosa, is highly susceptible to BD infection. When BD invades a naive site, these frogs quickly become infected and mass die-offs occur. On the other hand, the co-occurring Pacific chorus frog, Pseudacorus regilla, is much less susceptible to BD. Pseudacorus regilla can carry high infection loads without showing any signs of chytridiomycosis, and for this reason have been proposed as a reservoir host. How is it that these two species can have such a disparity in survival? Amphibian skin hosts communities of symbiotic bacteria, known as the skin microbiome. Recent research indicates that for some amphibian species, the skin microbiome may facilitate survival against BD by producing antifungal compounds or outcompeting pathogens for space. My research will compare Rana muscosa and Pseudacorus regilla skin microbial communities to determine how microbiome compositions may influence disease outcomes for these two frog species in the Sierra Nevada. To do this, I will visit 15 sites in the Sierra Nevada with different amphibian compositions and collect non-intrusive BD and skin microbiome swab samples from individual frogs. I will extract DNA from those samples and quantify BD infection intensities using a qPCR assay. I will use next-gen sequencing techniques to sequence bacterial DNA on frog skin and use bioinformatics tools to cluster, identify, and reconstruct the composition of these microbial communities. I will calculate diversity metrics and use visualizations to identify compositional differences between microbiome communities. And using multivariate analyses and mixed linear models, I will explore relationships between bacterial taxa, amphibian hosts, sites, and BD loads. My project aims to understand the role of symbiotic microbial communities in disease susceptibility and resilience, especially among co-occurring hosts. Understanding these factors will help inform conservation strategies for disease-susceptible species and hopefully move us towards better disease mitigation and amphibian recovery methods in the future. Thanks for listening.